In this first lesson on membrane transport, we're going to talk about diffusion. Here are your objectives. Let's start by defining active and passive transport. Active transport is transport across a membrane, moving a substance across a membrane that requires energy. Whereas passive transport is moving a substance across a membrane that doesn't require energy. There's no input of energy in passive transport. So why the difference in the energy requirements? Well, passive transport is taking things from high to low concentration. That's the direction that they're going to want to move naturally. Whereas active transport is doing the reverse. It's taking things from low to high concentration, and that's energetically unfavorable. I'm going to use these square brackets to mean concentration. That's the symbol borrowed from chemistry. To illustrate, let's forget about membranes for a moment and just think of a jar of water. If we were to drop a drop of red food coloring in the center, over time we would see that food coloring spread out to all corners of the water so that it became evenly colored. And that's diffusion. Diffusion is a process whereby the molecules spread out, and that happens because of their random motion. Now, diffusion can also happen across a membrane. There's no membrane here, but it can happen across a membrane. Similarly, with no input of energy and moving substances from high to low concentration. And that then, if it's across a membrane, would be considered passive transport. Diffusion across a membrane is passive transport. Whereas active transport would be the reverse process if we were taking the evenly spread out food coloring and moving it from low concentration to high concentration, and you can see that that would not tend to happen without an input of energy. So that's an introduction to active and passive transport. Let's talk further about diffusion. So the first kind of diffusion we can consider is simple diffusion. Again, we're moving things from high to low concentration. Another way of saying that we will use frequently is that we are moving substances down their concentration gradient. That means that we are moving them from high to low concentration. It's another way of saying the same thing. Now, across the phospholipid bilayer, PL for phospholipid, this kind of diffusion, moving things from high to low concentration, going straight through the bilayer, works best for things that can dissolve in the hydrophobic interior of the bilayer. So here's a phospholipid bilayer. And recall that the fatty acid tails that extend into the middle are hydrophobic, and they present a barrier to diffusion. So things that are going to be able to get straight through, this is what simple diffusion would be, going straight through the phospholipid bilayer, that's going to work best for things that are nonpolar and small. They'll be able to fit through and they will be able to dissolve in the nonpolar interior, whereas polar molecules will not be able to dissolve in this hydrophobic interior, and larger molecules will get stuck as well. So simple diffusion going straight through the phospholipid bilayer is best for things that are small and nonpolar. For example, small hydrocarbons. I'm going to draw just a chain of carbons and hydrogens like that. They can diffuse straight through. Carbon dioxide, which is small and nonpolar. Molecular oxygen, similarly small and nonpolar. Those things go through well. Things that go through poorly include ions, because ions are charged and they're not going to be able to diffuse or dissolve in the interior, the hydrophobic interior of the cell membrane. Polar molecules as well are less able to get through. Better than ions, but not as good as these. So things like water, which is polar, and glucose. So this is kind of a spectrum from gets through well to gets through poorly. Small hydrophobic molecules, small nonpolar molecules get through well, ions get through not well at all, and small polar molecules or larger polar molecules like glucose get through kind of in an intermediate way. That's simple diffusion, diffusion straight through the phospholipid bilayer. Now, facilitated diffusion is still diffusion, so it's still going across the membrane, and it's still not requiring energy. There's still no input of energy required. It's still going from taking things from high concentration to low concentration. Still a form of passive transport. The difference in facilitated diffusion is that it's providing an alternative route for things that can't get straight through the membrane. For example, I'm just going to draw my phospholipid bilayer like this. I'm going to draw these two lines. Imagine that there's more phospholipids here. This is a cross-section of the phospholipid bilayer with an integral protein in it that's kind of a donut shape. So I'll draw this from the side, and then I'll draw it from the top as well. This is the top view down here, and this is the side view up here. And again, this part here is the phospholipid bilayer. So a channel protein might look like this, and a channel protein provides a channel for things that can't dissolve in the phospholipid bilayer to sneak through. So for example, an ion like sodium could dissolve in water and then take this channel from the outside to the inside of a cell. That means that this part here of the protein, just like the outsides, is actually hydrophilic, not hydrophobic. And because it's hydrophilic, water can form a channel in here, and sodium can pass through as well. So this part is a hydrophilic core. Seen here, it would be this interior region would be a hydrophilic core. So channel proteins provide a hydrophilic core that ions and other polar molecules can use to get from one side of the cell membrane to another. This is an example of facilitated diffusion. Again, the sodium is still going from high concentration to low concentration. It's not being pumped uphill. It's still going from high to low concentration, but it's being provided an alternative route instead of going through the phospholipid bilayer. So examples of things that work this way, what I've drawn for sodium is an example of an ion channel. Water can actually move this way as well, and that's given the specific name of an aquaporin. Okay, so channel proteins, a form of facilitated diffusion. Again, still high to low concentration, but through a hydrophilic core. Another possibility is a carrier protein. Here's my phospholipid bilayer again. Here's another integral protein, but this one doesn't have a channel through it. What it does is it picks up a molecule on this side. So let's say there are molecules out here. It picks up a molecule on this side. These molecules are in high concentration on the one side and in low concentration on the other, and it actually carries it through, changing shape, changing conformation, and spits it out on the other side. So the protein itself is changing shape. That's different from this, where there's just a hydrophilic core. An example of a molecule that moves through a carrier protein is glucose. Again, passive transport going from high concentration on the outside to low concentration here, but because it couldn't diffuse straight through the phospholipid bilayer, because it is larger and it's polar, it takes an alternative route. Okay, so that's an introduction to facilitated diffusion and the two different kinds of proteins, channel proteins and carrier proteins, that are involved in it.